Morning children, and welcome back to the biology class. We are continuing with the lesson Biotechnology, Principles and Processes, right? So last class we saw that how restriction endonuclease or restriction enzymes are used to cut the DNA at specific points. Okay, now we can separate these cut strands, cut DNA by using a process called as gel electrophoresis. What is gel electrophoresis, children? Gel electrophoresis is a migration of undigested as well as digested set of DNA. Digested and undigested means if those got cut means it is got digested, right? Undigested means not cut. Okay, so we can be using more than one restriction endonuclease for cutting a DNA segment into small pieces, right? And whether this is proceeding, this is happening, we can know by subjecting the same DNA through an gel electrophoresis. Okay, what is this gel electrophoresis? That is, purified DNA is incubated with the restriction enzymes. As a result, DNA digests. DNA fragments are separated by a technique called as gel electrophoresis. Now, in this gel electrophoresis, a kind of liquid-like medium. A medium is used, which is got from agar-agar, or we can call it as agarose gel. Agarose gel is used for electrophoresis, is employed to check the progression of a restriction enzyme digestion. Okay, now you know that DNA is negatively charged particle. DNA is negatively charged and it moves towards anode. Okay, this gel electrophoresis is subjected to anode and positive. One of the terminal is positive terminal and the other is the negative terminal. Okay, and so it moves towards the anode. Now DNA fragments are separated according to their size. See, it is getting separated here, isn't it? You can see it is getting separated. How does it move in this agarose gel? See, these are the wells in which the DNA is placed. Cut DNA or the DNA which has got cut is placed. Okay, and it flows slowly, flows through this agarose gel towards the anode. This is the anode region, right? This is the cathode region. This region is the cathode region. This is the anode position. So this DNA molecule starts moving towards the anode. How does it move now? The smaller particles, smaller DNA pieces, which is having less density. The smaller DNA, which has the lesser densities, moves faster. You know that, right? So it starts moving towards the anode. Okay, now we can, if you want this piece, if I want this piece, the small piece of DNA, for using it for making recombinant DNA, suppose it is like that. Okay, I can cut it out from this agarose gel medium. Understood? Okay. Now DNA fragments are separated according to their size through sieving effect of agarose gel. Smaller sized fragments move further. This process is repeated with the vector DNA also. You know that the same kind of restriction enzymes are used for the recombinant organism as well as the vector organism also. Then only it will leave correct sticky ends. The same sticky ends it has to produce or leave. Okay, right. As the process is proceeding, the gel electrophoresis is proceeding, if they are subjected to UV radiation, you can see these like band-like things present, right? Okay, now I told this one, before visualizing in the UV autoradiography, you should be adding a chemical called as ethidium bromide. What is the name of the chemical? A dye. It's a dye, children. It is called as ethidium bromide. And then you subject it to UV radiation. Then you can see this. What is this? This band-like structures. Okay. Now, as I told you, I can separate this one of the DNA bands. DNA band this DNA segment. Whatever segment I want, I can, for using it for recombinant DNA, I can cut it out from the agarose gel by a process called as elution. What is the process called? Elution. DNA bands are cut out from the agarose gel by a process called as elution. Okay. Now you can see this one is the cutout gene, which is called as the gene of interest, which can be used to make a recombinant DNA in case of a plasmid. This is a plasmid. It's a plasmid. Okay. Right. What is the process called as? What is elution, children? It is the process of separation of this DNA fragments from the agarose gel by a process called as elution. Now I got a small piece. Okay, whichever piece I need, I have taken it out. I need just one piece only I got. I need hundreds and hundreds of copies of the same gene. What I should be doing now? Yes, the next process is, what we have to do is, next is the PCR test. It's not a test, it's a process. Okay, what is PCR? Anyone? PCR means polymerase chain reaction. What is PCR? PCR is the process of making thousands and thousands and thousands of copies of the same small segment of DNA. Okay, by using again the enzyme DNA polymerase. Okay, right. Now you might have heard of this PCR very lately. The test which is used to detect the antibodies in the patients of suffering from COVID. Okay. So what is this PCR actually? It is a polymerase chain reaction. And why it is done? To make not even hundreds, lakhs and lakhs of copies of the same gene can be done. Okay. Now what are the steps in the process of PCR? It is first the double-stranded DNA. DS is double-stranded DNA has to be split into. We are making millions of copies, children. So first it has to split into two DNA, right? Two templates, right? Like our actual replication, it has to get split. Now that process is done by providing heat. When heat is provided, it just splits into two strands, which process is called denaturation. Okay, denaturation is the first step. Second process is annealing. Now what is annealing? Now you know this DNA polymerase, the enzyme which will synthesize the DNA from 3' to 5'. So that's happening, of course. But before that, there's a small step where a small segment of oligonucleotide. What is that? A primer. Actually, it's a primer, or primer is actually an oligonucleotide. It's an artificially synthesized small segment of DNA, right? Okay, that is added at the 3' end. At the 3' end. This is the 3' end of the segment, and this is the 3' end of the segment. Okay, at the starting point it is added. Then the enzyme. What is that enzyme? DNA polymerase. DNA polymerase is naturally found inside our living organisms. But children, this PCR is done outside the body. So where do we get this PCR from? Sorry, not PCR. Where do you get this enzyme from? Polymerase from? Yeah, it is obtained from a bacteria called as Thermus aquaticus. What is the name of the bacteria? Thermus aquaticus. So it is called as TAC. TACQ. TAC. Thermus from the Thermus aquaticus it is taken from, right? Okay. So here it is added along with this entire thing is added along with the process as the process is continuing, children. So this, what is the specialty of this enzyme is? Enzymes are proteins, you know that. Okay. So but this enzyme, Taq polymerase, do not get destroyed at high temperature. We are using during denaturation, right? We are using high temperature for denaturation. At this high temperature, this Taq polymerase does not get denatured. Okay, that's the importance of this one. Okay, so this enzyme Taq polymerase helps to synthesize. This gets elongated like this. This gets elongated by, and so a new set of DNA is being synthesized. Okay, and like this, many after repetition of many cycles, millions of copies of the same segment of DNA can be synthesized. Okay, now this second step is called as annealing, where a small segment of primer oligonucleotide is added at the 3' end of each of this strand. That is annealing. And this next step is what? Extension using Taq polymerase. So children, you have to draw this diagram in your notebook also. Okay, so this is the repetition of cycles of the same PCR method to get millions of copies. Okay, right. So Thermus Taq polymerase from Thermus aquaticus. These are all the 3 steps is separately mentioned here. You please go through this. Okay, it's a very important question. Definitely it may be asked this year. Okay, because it is PCR. Okay.